Excellence in Science, Shimazu.
Hello everyone, welcome to Shimatsu. I'm Joel Langford, Product Specialist in X-Ray Spectroscopy at Shimatsu Scientific Instruments. I'll be your presenter, and today our topic is X-ray fluorescence spectroscopy for elemental analysis in pharmaceuticals. Before we start, I wanted to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The video will appear in the center of the screen. Directly under the video, you will see a resource list with clickable links relevant to the material being presented today. On the top left is the widget for questions and answers. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the Q&A box is the Request Information and Demo box. You may expand the items here to learn more about us. On the right are survey questions that you may fill out anytime during or after the presentation. Finally, at the bottom pane are the icons to bring up all these widgets in case they are minimized or hidden. All right, without further ado, let's get started. Again, if you are just joining us, I'm Joel, your moderator and speaker. Today, I will be talking about X-ray fluorescence spectroscopy for elemental analysis in pharmaceuticals.
So just a brief outline. I'm going to be providing a very brief introduction into X-ray fluorescence. And then afterwards, we will be discussing 4 pharmaceutical-centric X-ray fluorescence applications. And then I'll finally conclude. So bear with me a moment. Maybe to some members of the audience that this material is rather familiar, but it helps to understand at least the basics of X-ray fluorescence. So X-ray fluorescence is an umbrella term. Let me start by saying that we need to be a little particular with vocabulary here. So X-ray fluorescence is an umbrella term that means that encapsulates both energy-dispersive X-ray fluorescence, also known as EDX, and wavelength-dispersive X-ray fluorescence, WDXRF. In the context of this talk, I'll be interchanging vocabulary with XRF and EDX, but in the context of this talk, XRF and EDX are the same thing. Okay, so how does EDX work? What you do is you have an X-ray tube that emits a polychromatic distribution of X-rays, and those, for lack of a better word, those X-rays impinge. They're incident on the sample. And the X-rays electronically excite the atoms within the sample, which are composed of different elements. And there's a process of electronic transitions, particularly intershell electronic transitions, that are occurring. When the atom is excited by the X-ray beam, the primary X-ray beam, the incident X-ray beam, the excited atoms must relax. And this electronic relaxation results in the emission of what is called a secondary X-ray, or also known as a fluorescent X-ray. They're secondary because they're the second X-ray coming from the primary X-ray, but they're also called fluorescent X-rays. Because these electronic relaxations are discrete, or basically energy orbitals, electronic orbitals within atoms are discrete, each X-ray fluorescent photon has its own characteristic energy that is dependent upon which the element it came from. So for example, there's a particular energy like 1.487 keV for aluminum atoms, one for chromium and one for lead. So what you can do is use an energy discriminating detector, like a silicon drift detector, to obtain a energy dispersed X-ray fluorescence spectrum, which you see pictured here in a cartoon on the right. So in an energy dispersive X-ray fluorescence spectrum, on the horizontal axis, you have energy, photon energy, or X-ray fluorescent energy, which is normally denominated in units of kiloelectron volt. And so, for example, aluminum has nominally a emission energy of 1.487 keV. Chromium is at 5.4 keV, and so forth. So what you can do is identify the atom, identify the element from its emission energy, and its intensity is correlated with the concentration of that element within the sample. So roughly speaking, the higher the aluminum intensity, the more aluminum content is in the sample, and vice versa. This is a picture of a Shimatsu Energy-Dispersive X-Ray Fluorescence Spectrometer. It's called an EDX7000. It's composed of a 50-watt rhodium X-ray tube, followed by a set of filters, which I'll mention in a little bit, a collimator, which is used to choke down the X-ray exposure area. So for example, if you have a inhomogeneous sample and you want to analyze a particular area on it, you can use the collimator to choke down the X-ray beam on that particular area. A CMOS camera, which is used to take pictures as well as align the X-ray exposure area to a particular part on the sample, as well as an energy discriminating X-ray detector that we call a silicon drift detector. What really sets us apart is the filters. And without getting too technical in detail of the science of the filters are really important in pharmaceutical analysis. What they essentially do is they remove, decrease background that is resultant from primarily Compton scattering. Some people call it the anomalous scattering, but primarily X-ray scattering off of the sample, and thereby decrease detection limits. So there's a filter for a particular element. For example, there's one filter for lead and mercury. There's a filter for sulfur. There's a filter for arsenic, and so forth. But essentially, to summarize, these filters are used to increase signal to noise by eliminating or reducing background that comes from Compton scattering that is actually rather prevalent from organic matrices. Shimatsu's XRF systems can be equipped with an autosampler. So the standard autosampler is a 12-position autosampler. It's designed to fit a variety of XRF cups. Now, what's really nice about the autosampler is that you can customize an analysis cue or spool to any fashion you want. For example, you might put multiple samples in there where one set of samples you're interested, the target elements might be catalyst-like elements like palladium and platinum, where another sample might be more, the target elements might be the big four like arsenic and lead. So you do not have to come to the instrument if you need to run a different target element method. You can simply spool up the methods with our user-friendly PCDX software, PC Energy-Dispersive X-Ray Fluorescence software and simply hit go, and the instrument will do the rest. So these come from our customers. These are just some of the benefits of using EDX. And in particular, when I enumerate these and describe them, it's good to have the background of other elemental techniques like atomic absorption and ICP-MS in the back of your mind. So one of the benefits is that there is no need for the analyst to spend long periods of time working in front of the instrument. The analysis may be completed in a short period of time as well. Analysis can be anywhere between seconds to minutes to an hour, depending on the element, the detection limits that are necessary. But regardless of the analysis length, the analyst does not need to be in front of the instrument while it is measuring the sample. It is easy to measure some elements that are difficult to measure by ICP-MS. These are primarily the halogens like chlorine and bromine, but also other elements like sulfur and phosphorus. There's no need to make a calibration curve every time. Curves last for a very long time, sometimes months, on an XRF system. Rapid startup and minimal preparation equipment. It just takes minutes to turn on an energy-dispersive X-ray fluorescence analyzer, and there's no need for mobile phases. And for example, a technique like ion chromatography or another liquid-based chromatography technique. There's no waste after analysis. This is very key, actually. For example, techniques that use mobile phase accumulate liquid waste, but XRF is a non-destructive technique, meaning that you recover the sample after analyzing it. And this can be very important in, for example, a research and development or a research and discovery type environment where basically very small amounts of material are being synthesized. So having a non-destructive technique for elemental analysis is a big plus when it comes to these environments, like research and discovery environments. A small amount can be analyzed as little as 0.1 grams. Many elements from sodium to uranium can be analyzed in a simultaneous fashion. You don't have to worry too much about contamination, and the running costs are low. The main consumable cost is actually electricity, which is equivalent to that of like a desktop computer. Because for the rest of the talk, I'm going to be comparing a lot of EDX with ICP-based techniques like ICP-MS and ICP. So it's worthwhile making a chart that describes on a basis-to-basis fashion the differences between EDX and ICP-MS. So in EDX, a sample matrix can be a variety of materials. It could be a liquid, it could be a solid, or it could be a powder. In ICP-MS, oftentimes APIs are powders. And in order to measure the API in an ICP-MS, it needs to be converted into a clean, slightly acidified liquid. So ICP-MS, you need to have a liquid to measure it. EDX can accept a variety of matrices. The sensitivity, this is often a critique for EDX, that the sensitivity is too low for elemental analysis, and I hope to show you that's not the case for the rest of the talk. Nominally speaking, the sensitivity of an EDX is in the low part per million to very high part per billion region, whereas an ICP-MS is part per trillion. Now, the nice thing is the dynamic range of the EDX is very high. It's part per billion, 500 part per billion, all the way to 100%. Now, the instrument sensitivity this is true, part per trillion levels for ICP-MS and part per million levels for EDX. However, oftentimes the method sensitivity for ICP-MS, especially for pharmaceutical analysis, is on the same scale as EDX, because if you're taking a powder, you have to digest it and dilute it by anywhere between a factor of 100 to 1,000. So this dilution process results in a method detection limit that's on par with an EDXRF method. We're going to be talking a lot about pharmacopoeias in this talk. USP 735 is a chapter in the pharmacopoeia that is specific for X-ray fluorescence analysis of pharmaceuticals. 233 is that for ICP-MS and ICP. So what you can think of how I like to think of USP 735 is USP 735 allows the user to consider using X-ray fluorescence in place of that used for ICP-MS by USP 233. The pre-treatment for EDX is very simple. You just simply put the powder in a XRF container. ICP-MS is very complex. You got to dissolve using something like a microwave digestion system with acids, and you need to dilute as needed. The measurement time for EDX, particularly for impurities in pharmaceuticals, is what I quote as long. Now, it could be like an hour long, whereas the ICP-MS, the measurement time is short, seconds to minutes. However, when you consider the whole entire method, which includes the digestion and the dilution process, oftentimes the throughput of an XRF method is actually higher than that of an ICP-MS. There's really nominally no equipment for EDX. There could be a draft for powders. But for ICP-MS, you're working with a lot of acids. You're working with argon dewars or cylinders. There needs to be acid-resistant glassware. You could be sometimes working with hydrofluoric acids, for example, for pharmaceuticals where titania is the excipient. And then on a consumable basis, the instrument, the EDX has very little consumables. You can measure all the elements in atmosphere. And for ICP-MS, argon is necessary. Also, on an electricity basis, EDX is much more frugal compared to that of ICP-MS. This is the periodic table. So the International Center for Harmonization, Q3D guidelines, they specify different classes of elements that need to be essentially in the peripheral of, they need to be considered for analysis in pharmaceutical matrices. So there's the class 1 elements. These are notoriously called the big four, like cadmium, mercury, lead, and arsenic. Class 2 elements, the class 2B elements, which are the catalyst-centric elements like rhodium and palladium, and the class 3 elements. All these classes can be analyzed by XRF except for lithium. This is an example of a workup, a basic workup, a basic flow of how you would prepare a powder sample, something like an API for XRF analysis. So you might have an API powder, you put it into a dedicated XRF container, and this powder is supported with normally it's a polypropylene film. You might tap the powder or tap the cup to distribute the powder within the cup, and then you simply set on the instrument and press the go button. So this saves a lot of time and costs, especially compared to methods like ICP-MS and ICP that oftentimes require digestion and dilution and workup with acids. Okay, so we're going to go into some data here. And what I want to emphasize is that all the data is coming from marketing materials that Shimadzu has previously published. So I'm just showing two of the application news or application notes that have some of the data that I'm about to show, but there's actually quite a few application notes out in the ether, as well as some white papers, as well as some manuals, a lot of marketing material. If you're interested in any of these documents, please feel free to contact us, myself, or any of your nearest Shimadzu sales representatives. Okay, so this is a synopsis of some of the applications I'm going to show. Now, XRF is, in the United States, is, in fact, in the world per se, is a massive market. And XRF is used in a variety of industries just outside of pharmaceuticals alone. For example, in agriculture and geological applications like mining and cosmetics and OEM and so forth. So, and even within the pharmaceutical industry, there's more applications than what I'm just showing today. So the first application is identifying catalyst residues in active pharmaceutical ingredients. The next application is, I actually think is pretty cool. It's pretty interesting. It's something that it's pretty, I don't want to call it novel, but let's just say, I'll show it later, of course. It's a very interesting application, and that's chlorine chloride analysis in API. And then application 3 and 4 are more, for lack of better words, are more nitty gritty and more USP centric, where I will be using some vocabulary that comes from USP 232, 233, and 735. And this actually, application 3 and 4 covers not only APIs, but a matrix that would be in a solid dosage form, like for example, a tablet or a pill. Okay, so the first application, identification of catalyst residues in API. So this is from one of our pharmaceutical customers. We'll keep them anonymous. It's in the analytical and research department. And what they're using the EDX for is the analysis of catalyst residues in APIs and different intermediates along the production process to make the API. They also use, it for elemental purity analysis in APIs, things like lead, arsenic, class 1 elements. The reason they chose the EDX system was that the target elements, they were primarily interested in osmium and palladium, had a good correlation with that obtained from ICP-MS. And I don't cover it in this talk, but the other reason they chose our system is that Shimadzu offers an EDXIR analysis software package. And please expect future webinars about this. But the EDXIR analysis software package it's a great quality control tool because it allows for the analysis of both organic and inorganic impurities, contaminants, foreign matter, for example, by combining the capabilities of both XRF, EDX, and FTIR. Okay, so here's the API. It was benazepril hydrochloride. Okay, so this is the example workflow, you had the API, vinazopyryl hydrochloride, that we call the drug substance. We spiked it with ICP standards, the 5 elements you see, iridium, platinum, rhodium, etc., each at 50 ppm. We mix that spike in a mixer mill, put it in a dedicated container, and we simply hit the go button. We put it in the EDX and hit the go button. We also use an unspiked sample as a control and to compare, for example, specificity and selectivity. Now notice that we're bypassing a massive step. If you wanted to do elemental impurity analysis by ICP-MS, you would need to use a microwave digestion system with all the reagents and consumables associated with that. Okay, so Shimadzu offers a software package that is named, for lack of better words, the Pharmaceutical Impurities Analysis Method Package. All the data that I'm going to show you was collected using this methods package. And it primarily goes over, it covers the class 1 and class 2A and 2B ICHQ3D elements. What's nice about this method package is it contains methods and suggested standards that you should use. In this case, it's a mixed standard solution manufactured by Specs that really reduces the method development process. Without getting too much into the science of this software package, there's certain correction factors that are used. For example, overlap corrections, what's called a ratio method, a Compton background correction, that are used to better improve the quality of the data. Now, that being said, you can do this without the method package. However, you will have to develop your own methods. What this method package does is it provides a shortcut and allows the user to not question interferences, basically. It really, in the end, shortens down the method development process. This chart compares, essentially is a comparison between EDX and ICP-MS. So we have 2 APIs, vinazopyryl hydrochloride and captopril, and we spiked it, and with the EDX 7000, we got 50 ppm plus or minus, let's say. Well, the RSD is there, but the plus or minus 5 ppm or so. And this is compared to the ICP-MS, which also got 50 ppm. The RSD is as measured by putting multiple replicates inside an EDX. This is not from, you don't calculate the RSD from the same sample, but the same sample in different cups. And the RSD is less than, to be frank, is very good. It's less than 2%, which is, as you'll see, meets the USP specification. So in the end, the results between the EDX 7000 and the ICP-MS are nominally very similar. This is similar to, I call this a robustness criteria. And what this is it's calculating the rate of change as a function of sample weight or sample volume. And so what we have is nominally 2 grams is the standard sample volume that's put in the cup. And then we prepared other cups with 1 gram, 0.5 grams, and 0.3 grams. And what's nice is that you don't see a systematic change in the change rate according to the spike sample volume, according to the spike sample weight. Now, why is this important? In a manufacturing setting where you have different users, for example, that have different, for lack of a better word, different tendencies, this is part of the proof showing that the method is very robust. This is examples of analysis at very low, and what we call low and very low. That's kind of a subjective term, but low and very low concentrations, where we basically took different sample volumes, but spiked with 10 ppm and 5 ppm standards into benazepril hydrochloride and captopril. And you can see that regardless of the sample volume, there's no rate of change, first of all. There's no systematic rate of change. And the recovery is, for lack of better words, is rather high. Some of the elements are not included in the pharmaceutical analysis method package. The class 1 and the class 2A and some of the class 2B elements are, but there's a few that are not. Now, that being said, that does not limit you from doing elemental analysis of those elements in pharmaceutical impurities or pharmaceutical matrices. Okay, so for example, here's osmium and tin, class 2B and class 3 elements according to the ICH Q3D guidelines. And this data shows it was done by spiking in cellulose and using that spike and quantifying it against an aqueous solution calibration curve. And you can see that the quantitative values are, for lack of a better word, are fairly accurate, with a 10 sigma value that's less than 1 part per million, at least for the osmium case. Now, there are other elements that are also outside of the ICH Q3D guidelines, things like silver, gold, thallium, chromium, so forth, molybdenum, and so forth. These can all be analyzed in principle in pharmaceutical matrices, in pharmaceutical APIs, for example. If you have questions with regards to the analysis of these elements, please feel free to contact Shimatsu. We will be happy to help and develop methods with you in contingent, in a how do you say, in a helpful fashion. Okay, this is an interesting application that's revolved around chloride, technically, not chlorine, but chlorine analysis in APIs. So this is a pharmaceutical customer that we will not name. It was a drug discovery technology research institute. And they were actually, the main reason that they purchased an EDX was actually to replace ion chromatography. They were using ion chromatography to quantify chloride and benazepril hydrochloride. And basically the target elements, the palladium, chloride, and tin, had a really good correlation with the quantitative values obtained from ion chromatography and ICP-MS. So another API, the same. Once again, we encounter benazepril hydrochloride, and we put a very small amount of sample, 200 milligrams, into a container. It's called a micro XL container. It's a container meant specifically to deal with low volume samples. And once again, we set in the instrument, we analyze an atmosphere for about 5 to 15 minutes. Okay, in the box here, you can see that we were able to quantify chloride, to a value of 7.51%, where the theoretical value of chloride, as calculated using the molecular formula of benazepril hydrochloride, is 7.69%. Now, you might be thinking, how is this even possible without the use of external standards? And what's nice about the X-ray community in general is that they enjoy theoretically interpreting XRF and actually all sorts of X-ray data, X-ray absorption, X-ray photoelectron spectroscopy, X-ray fluorescence, Sachs and wax data, and so forth. And there's actually a theory out there for quantifying elements within matrices without the use of external standards. And this method of quantification is called the fundamental parameter method. This method of quantification, there's some intricacies into it, and it involves a little bit of math like matrix algebra and the process of normalization, the mathematics of normalization, and an algorithm. But all of that comes as standard with the EDX7000 software. So fundamental parameters can easily be used to quantify data using Shimatsu's EDX7000 system. Now, to prove that this wasn't just a one-off case, we measured many different APIs: benazepril hydrochloride, propanol hydrochloride, bromhexene hydrochloride, and so forth. In the green rows, you see the value quantified by EDX using fundamental parameters. And in the white is the theoretical value as calculated from the molecular formula. And you can tell that the value is within nominally 1% of the true value. And so for applications that are, for example, a screening application where it's not incredibly important that part per million type levels need to be known, this method works. And so this customer actually uses this fundamental parameter method as a screening tool to check that the chlorine value or chloride value is within a nominal or a sensible judgment range, so that it can be so that the product can be further assembled into, for example, a drug like a tablet or a pill. Okay, so now we're going to be talking a little bit about the intricacies between XRF and the pharmacopoeia chapters. So these two last methods revolve around elemental impurity of APIs and solid dosage forms, with the backdrop of the pharmacopoeia chapters in the back of our head. So I'm going to be talking about two customers that use the XRF system to meet specifications within the USP guidelines. And so one is, we're both leaving them nameless. One is a medicinal chemistry research institute. They use it to identify catalyst residues during the early on stage of catalyst purification. The target elements, lead, class 1 elements, were found to have good correlation with their ICP atomic emission systems. And the nice thing is that the EDX7000 actually had a higher throughput because of the lack of complicated sample preparation as compared to that of the ICP method. The second customer is in a pharmaceutical research institute, and they were using the EDX for catalyst residues during scale-up research for API production. They were interested in 8 different target elements that had good correlation with ICP AES. And actually, the big reason they bought this was the EDX 7000 can be made 21 CFR Part 11 compliant. And I don't talk about it in today's talk, but it's not an issue at all for Shimadzu's EDX 7000 system to be made FDA 21 CFR Part 11 compliant. For more information about that, please contact us and we'll be happy to discuss the intricacies of making this instrument into a 21 CFR Part 11 state. It's necessary for manufacturing settings that require electronic security, traceability, and so forth, in a high quality control environment, for example. So we're going to be covering a little bit of vocabulary here, and sometimes from my perspective it's easy to get lost in this vocabulary. These are the vocabulary that's specific to USP 735. So if I say something slightly incorrect, please forgive me. Okay, I made this chart and highlighted the crux of it in yellow. So there's a criteria for linearity, which is basically a regression value of no less than 0.99. An accuracy criteria, which you take a spike sample, and as long as it's within a 70 to 150% recovery, then you've met the acceptance criteria. The specificity, which I actually find to be the most interesting criteria, it's basically in USP 735, it is met if you meet the accuracy criteria. And I'll explain why that is in the next few slides. Repeatability, which is measured by measuring replicates, and that the replicates do not have a standard deviation of no more than 20%. The quantitation limit, which is to get an LOQ in USP 735, you measure a blank at least 6 times, no less than 6 times, and you take the standard deviation and multiply it by 10. There's another quantitation limit that I'm going to be talking about in the fact that the analytical procedure should be capable of determining the analyte precisely and accurately at a level up to 50% of the specification. I'm going to be talking quite a bit about primarily the word specification and what it means in the context of USP 735. And finally, the robustness. I have for a lack of better words, is can you deliberately change the experimental settings or basically screw around with the instrument such that the change rate is very low or within plus or minus 20%. Now, what constitutes a deliberate change? These are things like using 2 different EDXs, using different analysts, using different sample volumes, maybe using a different sample cup, maybe using a different film. Those are deliberate changes within the experimental parameters. For more information, for more of the intricacies of USP 735, I suggest please refer to the original USP 735 text. I should mention that USP 735 was adopted rather recently. It was adopted in 2018. Okay, so here's an API. It's captopril and benezopril hydrochloride. The daily amount of the drug is 10 milligrams per day and 150 milligrams per day. And these were the standard samples used to make the curve. We had a blank, and then the standards were provided by specs. They're called XSTC 2046, and we made 4 different standards by the method of dilution. So for example, dilution ratio is 15 ppm, then of 2 is 7.5 ppm for arsenic, by 5 is 3, etc. Okay. So in USP 232, they describe the permitted daily exposure values according to an oral administration route. There's different administration routes. Another one, for example, would be inhalation. We're going to be discussing oral, the method of oral introduction, like from a tablet. So the permitted daily exposure values for the class 1 and class 2A elements are listed in the top row. 15 ppm for arsenic, 30 microgram, sorry, not ppm, actually 15 micrograms per day of arsenic, 30 micrograms per day of mercury, 5 micrograms per day of lead, and etc. Then the maximum permitted concentration. Okay, how do you get the maximum permitted concentration from the PDE values? There are different conversion factors specified within the USP guidelines. We used option 2B, which is listed below, conversion method to maximum permitted concentration. What this is, the idea behind it is, from my perspective, is that you have a drug and the maximum intake value of that drug is something like, the daily intake value of that drug is something like 300 milligrams, which is common for something like a tablet. Okay, so what you do is you take the PDE value and divide it by 0.3 grams, and then that gets you a concentration. So it would be 50 ppm of arsenic, 100 ppm, microgram per gram, which is the same as a ppm for mercury, and so forth. And then the target concentration is 30% of that value. So it'd be 15 pp. It would be 50 times 0.3 for arsenic, 100 times 0.3. That target concentration is the specification value that is said in the USP method. That's what I mentioned previously. What does specification mean? It means the target concentration. So half of the target concentration, half of the specification is 7.5 ppm for arsenic, 15 ppm for mercury, and so forth. So this will be the flow, very similar to what we saw previously. You take the powder, you have an unspiked sample, simply put it into a cup, tap it a little bit, and set it in the instrument and press go. For the spiked sample, you spike and mix the 7 elements. You have your spiked sample powder, put it in a container, hit the go button. The linearity criteria. Here's the data. For lack of better words, all the curves met the specified criteria, which is an R of no less than 0.99 for all the elements. So all for what we say here is the judgment passed in terms of linearity. Accuracy and precision. Okay, so what we have is we spiked the 2 APIs to half of the specification value. So in this case, mercury would be 15 and lead would be 2.5. Okay, and what we did was we quantified those. We also did multiple runs, so we have an RSD and a recovery rate. And we had to show that that recovery at half of the specified value was within 70 to 150%. And that also, the precision criteria is that the relative standard deviation is no more than 20. You can take a look from the data for both of the APIs, the accuracy and the precision criteria, for lack of better words, passed with flying colors. Specificity. Now, in USP 735, if you meet the accuracy requirement, you don't think about specificity because you've met it. So I'm going to talk about what specificity means. It's essentially the ability for the instrument to, you can think about it as if you spiked one element within this matrix, would that influence the content of the other elements? And because you're spiking for the accuracy specification, you've met the specificity argument, the specificity criteria. So for the specificity criteria XRF, the EDX 7000 meets the specificity and criteria because it met the accuracy requirement. Quantitation limit. Again, to get the quantitation limit, you measure a blank, for example, unspiked benazepril hydrochloride 10 times and multiply that by 10, and you get the values listed at the bottom here. Again, we spiked at 7.5 ppm because that's half of the spike. It's half of the specification. So 15 ppm, the spike concentration would be 7.5. It would be 15 ppm for mercury and so forth. And as seen here, the concentration determined from that spike, it was easily able to quantify the analyte precisely and accurately at a level equivalent to 50% of that specification. Robustness. In this case, very similar to what I showed previously. Changes in sample volume, do they systematically change the change rate? So for both of these APIs, the benazepril hydrochloride and captopril, we had to show that the change rate is not systematic with sample volume and it's no more than 20%. Again, this can be important in a manufacturing setting where throughput is very high. You want to show that the method is robust. And in this case, from the perspective of sample volume, from putting the powder into the cup, the method passes this criteria. For lack of better words, it is robust. Very similar to application 3, we're going to be talking a little bit about USP 233 now, which is the ICP USP method. We're going to be doing elemental purity analysis of essentially tablets or in the oral solid dosage form. So this could be something that has an API with a filler, excipients in it, things like cellulose, titanium, talc, etc. Okay. So this comes from the limit procedures in USP 233. So what you have is a cellulose powder, and you're supposed to spike it with respect to a parameter called J, which is the specification concentration. So for arsenic, it's 9 ppm. For mercury, it's 18 ppm. For lead, it's 3 ppm. Or 3 micrograms per gram. It's the same thing. So you have a blank, one spiked cellulose powder at 0.5 of J, one at 1.5 J, etc. Then you have the test samples, which one should be spiked at the target concentration at J, and the other should be spiked to within 80% of the target concentration, so 0.8 J. So there's two detectability parameters, two detectability criteria. So the detectability of sample powder 2, which is at 80% of the target concentration, you need to prove that that average value is always less for all the elements than the standard powder. And then the detectability for the sample powder 1, it should be plus or minus 15% of the average value of the standard cellulose powder. So that would be the plus or minus 15% within 1.0 J. There's also criteria for repeatability and specificity, like repeatability, relative standard deviation of less than 20%. So here's the spike sample powder 1 and the standard powder 1.0 J. So this is coming from a tablet where we're assuming the intake is 0.5 grams. So we used option 3 to convert the permitted daily exposure value to the maximum permitted concentration. So in this case, because it's 0.5 grams, last one I showed was 0.3 grams. What we would do is take the PDE value and divide that by 0.5 grams. The target concentration is 30% of that value, so it'd be 9. And then we spiked at that concentration. That's 1.0 J. So it'd be 9 ppm, 9 micrograms per gram of arsenic, 18 micrograms per gram of mercury, 3 micrograms per gram of lead, etc. So here's an example of a workup that would be something like a tablet. You pulverize it with a mortar or pestle or a mechanical grinder, for example. That would convert it into a powdered sample. And from on that point, you can treat it like essentially the previous API powders that I discussed. You can simply put the powder sample into a measurement cup and tap it, set it in the instrument. And very similar to the standard sample, the cellulose powder, you take the solid dosage form, pulverize it, and mix it with 8 spiked elements, put the spiked sample powder into a cup, and set it in the instrument. So the correlation curve for the 3-point curves of 0, 0.5J, and 1.5J, the correlation coefficient is above 0.99, which shows a good linearity, and it meets the specifications that are provided within USP 735, for example. Detectability criteria 1 and 2. So you have the standard power 1.0J. The top row is the quantified values using the 3-point curve, so 9 ppm for arsenic, 18.2. Then you have the spiked sample powder. Okay, so the standard powder was made using cellulose, and the spiked sample powder is a tablet that has like excipients, fillers, APIs in it, etc. And you can see that we meet within the criteria. We are within the criteria, I believe it's plus or minus 15% of the of 1.0J. And so it passed with flying colors. The spiked sample powder one passed with flying colors. Spiked sample powder 2 is 0.8 of the target concentration. And for all of the elements, we showed that the spiked sample powder 2 is always less than standard powder 1.0J. So it met this detectability requirement, detectability 2 as well. Specificity. So we can compare the spectra from like for example, a blank cellulose powder or spiked sample powder 1 and unspiked sample powder, and we can see that each element has its own individual peak that needs to be detected. And because we passed the detectability determination, the specificity was also passed. This is very similar to like we passed the specificity because we passed the accuracy criteria in USP 735. Repeatability. 6 samples of spiked sample powder 1 were prepared and set into a turret that you see here. And we calculated an RSD from that. And it met the plus or minus, forgive me, I believe it's 20%, no more than 20% RSD criteria. It passed very well within specifications. Conclusion. So I'd like to conclude with these following notes: that the importance of elemental impurity controls and elemental impurity control in medicines is increasing against the harmonization, the globalization of the world, the ICH Q3D guidelines. The EDX 7000 makes it easy and fast to check catalyst residues and elemental impurities in APIs. especially compared to a technique like ICP. We verified 2 different APIs against USP, the criteria specified in USP 735, and the method met that criteria, no problem. And then finally, we used a tablet, validation of an oral solid tablet in accordance with USP 233, the limit procedure specified within USP 233, and the acceptance criteria was met. I leave with this note: the importance of elemental impurity control in medicines is increasing against the backdrop of the release of ICH 23D guidelines. Shimatsu's EDX system offer a simple and fast solution for elemental analysis of medicines in accordance with these guidelines. I thank you for your time.
Under the resource list, you can find more information about our analytical solutions. If you have not had a chance, please fill out our survey questions on the right-hand side to provide feedback and request additional information. We also encourage you and your colleagues to attend our upcoming webinars, which encompass a wide variety of application areas.
Excellence in science. Shimazu.